Hi everyone. In this video, we are going to learn about the organization of DNA. So the different levels presenting in DNA organization from its DNA to chromosome structure. So when we take the organisms in the organism, also we see the different levels of organization. So the organization, like we see that cellular level to we see. Next is tissue level. From tissue level to we understand the organ level and organ system level. So when you see from the cellular level to the organ system level, you see the complexity in its structure. Similarly, when we take DNA structure from its when you take this genetic material from its DNA to the chromosome structure, we see the different level of organization, and we find the compact structure or the complex structure of this particular molecule. Okay, so coming to this one, okay, when you take the DNA, we know it's just a string of two just a molecule with two strings. We know that is just DNA is a molecule with two strings, and when you measure the diameter of these two strings, so the diameter is 2 nanometers. So the diameter is 2 nanometers, and we know that this string also binds with some protein molecules, and those proteins are called histone proteins. And so the histone proteins, they bind with these strings, and when the histone proteins they bind with these strings, we call it's a nucleosome. So DNA with this particular histone proteins together, we call it is a nucleosome structure. Okay, so coming to these histone proteins, the histone proteins are again two types. So core histone proteins and linker histone proteins. So these core histone proteins are 8 proteins complex. It's 8 proteins complex. So in these 8 proteins, so histone 2A proteins, a pair of histone 2A proteins, histone 2B proteins, and a pair of histone 3 proteins, and a pair of histone 4 proteins. So these 8 proteins we call is octamer complex. So this octamer complex forms core histones. And the linker histones, so they are just a H1 protein. It's a single it is a single protein. H1 protein is just a single protein, okay, is present. So the binding of so this proteins to these strings. So what advantage that gives to DNA by binding of this histone proteins to this DNA molecule? So we know that the DNA molecule is a very it's a very long string. So it's a long string and it's a linear form. So to accommodate this DNA molecule inside of the tiny nucleus is not possible. So to keep or to make sure to ensure that this DNA is present inside of the nucleus, okay, the DNA undergoes some structural changes. That means this string structure DNA becomes some changes, so that it becomes more compact, that can be accommodated, or that can be reside inside of the nucleus. Okay, so as I said, this 2 nanometer structure of DNA in its long form, in its long form condenses. So when it condenses, obviously what happens? It's the length shortens, and the diameter increases. The length shortens and the diameter increases. Now we are going to see how the diameter of this particular molecule increases from its from primary form to the advanced form, or it's from its starting form to the the last form. So you should understand that when the molecule when this molecule undergoes changes, we observe the changes in its thickness. We observe the changes in the thickness. How this particular string, which is in a very long in size, becomes a short one. So it is possible. How it is possible? So it is only possible when it folds back like this. So it may be possible for the to make this a long strand into very short form structure, or a condensed form structure, so that the long size molecule becomes a very short size molecule. But the organization, when you observe in this DNA, so definitely you will understand. So it's not just folding back of a long string, so that the long DNA molecule becomes short and forms a new structure. It's not such a simple thing. So when you see the DNA, as I said, no, it's just as two strings without any proteins associated to it. So when you see the structure of the nucleosome, there you see the core histone proteins. So this DNA molecule, so when you take the DNA molecule of this length, for example, just imagine 5 centimeters, and when you twine this DNA 5 centimeters of this length onto this particular string, okay, particular around these proteins, so what happens, this 5 centimeters one now can be accommodated in just in a 1 centimeter length. It can be accommodated in 1 centimeter length. So here you have to understand, so one thing, the DNA becomes short, and as well as you can see because of this twining, you can see the diameter of this particular one is increasing. The diameter of particular one is increasing because inside of this strand now the histone proteins are present. So the diameter is increasing. So the diameter increases from 2 nanometers to 10 nanometers now because of this twining and inside of these proteins, histone proteins, core histone proteins. Okay. And when you observe the DNA strand along its length, you find this kind of structures like this. Okay, so multiple such nucleosomes you can identify. So all these nucleosomes, so in between these nucleosomes you find some region still which is connecting these two nucleosomes. This we call it the linker DNA, and there are some proteins we call histone proteins only, H1 histone proteins. So they join or they bind to this particular region. That's why we call these H1 proteins as a linker histone proteins. They are called linker histone proteins. So the nucleosome with these H1 proteins, we call it a chromatosome. So chromatosome has both this core histone proteins, and also you can find histone protein. So now if you see the diameter of this particular string now with this both with this core histone proteins and this linker histones, though now the size of this particular string DNA string with this proteins is now 11 nanometer. Okay, so that means the DNA, so the length of the DNA reduces in this particular process, and also you see the thickness of the DNA also simultaneously increases in this particular process. So the next level one is chromatin fiber. So what is the chromatin fiber? So when such multiple chromatosomes, that means with linker proteins, with multiple such linker proteins and these nucleosomes, so this chromatosome forms chromatin fiber. What it forms? Chromatin fiber. So when you see in a structure like chromatin fiber, you can understand that, okay, the chromatin fiber has a multiple chromosome structures. Multiple chromosome structures means the DNA string with its core histone proteins and the linker DNA. So what is the advantage for DNA with these linker proteins that we have to understand now? Okay, we have these nucleosomes or chromatosomes. I can say, we have nucleosomes. Initially we have nucleosomes, and later the histone proteins binds to this one. So when the histone proteins binds to this one, so we call it a chromatosome. Okay, and because of these chromatosomes, and when such multiple chromatosomes are present, we call that is a chromatin fiber. So we call it as a chromatin fiber. When you see the structure of the chromatin fiber here, see this chromatin fiber, you may find that it's just a linear structure, like a you find this without this linker proteins. So when linker proteins are not there, still you can find this structure. So what these linker proteins further brings change in this particular DNA strings. So because of these linker proteins, what happens, now this DNA arranges in multiple layers. So you can see, because of this, the length of DNA, that means, so the DNA charts, the size of the DNA or the length of DNA, so reduces further, and also you see the thickness of the DNA increasing. Thickness of DNA is increasing. That means the fiber, the DNA fiber, the chromatin fiber thickness is increasing. When you see the diameter of this chromatin fiber, so the diameter of chromatin fiber, you see that the particular chromatin fiber size is 30 nanometers now. So what is the next level of organization in this particular chromosome organization or DNA organization? Okay, the DNA in this chromatin fiber, okay, so it forms a loops. So for example, when you imagine a cloth line, so where we use the clips, so just imagine this particular one. Okay, just imagine, like, no, I'm going to draw even clear. So suppose this is a, so when you just imagine, say, just is a, imagine it's a line, cloth line, and where we use the clips to hang the cloths. So the same way, when you take this chromatin fiber and hang over this scaffold structure, you find this kind of loops formation. So the chromatin fiber forms the loops further, and this compacts even the size. See, the size compacts. Okay, particularly when you take this one, so around this scaffold you find this continuously like this. Because of this, you will find the thickness of the DNA gets increased. So you find that the thickness of DNA gets increased, and also the length of the DNA string now reduces, or becomes even less when compared to its previous structure. And the chromatin in this loop structure will forms the chromosome, the next level. So when this particular chromatin fibers with these loops, when you see, this is a scaffold structure, and proteins which present to ensure that these chromatin fibers to join these scaffold structures are called cohesin proteins. Okay, and also we find some condensin proteins. So basically, the condensin proteins, they keep the these particular chromatin fibers to keep this particular chromatin fibers to join with, to ensure that, to make these chromatin fibers to join with this scaffold structure inside of this particular chromosome. So it's a non-histonous protein scaffold. Okay, so these cohesins are also very important. So they keep the DNA, so double strand DNA molecules, after replication, so they must be very close to each other, so that during the mitotic phase, so that the cells, they can form this chromatins. So to form this particular chromatin structure, so definitely this double stranded DNA molecules, they must be very close to each other. So to keep the molecules very close to each other, so this particular proteins, what I said, no, this cohesin proteins, so they keep the DNA strands together. So this is about the structure of DNA organization and its structure. So initially we find it's just a string structure, then we see the strings having the histone proteins, and when the histone proteins when the DNA twines around these histone proteins, we call it a bead structure. That's why we call it a beads on string. When you take this particular chromatin fiber, at the chromatin fiber level you can see that no multiple such beads are present on the string. Okay, that's why it's called a multiple such chromatosomes are present on the DNA, along the DNA. That's why we call say strings beads on the string structure. Okay, further this beads with string forms the loops, so that's finally this particular kind of structure loops, fiber chromatin fiber with loops will form the chromosome. Thank you.